Hi, everybody. So welcome to this lecture on linkage mapping, linkage analysis for as applied to human genetics. And so hopefully this lecture is being recorded just in case the live lecture doesn't work. And this recording is there as a backup for you to find if the live lecture doesn't work. So we're going to talk about linkage analysis, and in a way, linkage analysis is not really such a big deal in human genetics as it was 20 or 30 years ago, when it was kind of in its sort of heyday. But I think it's important to go through linkage analysis for kind of a few reasons. So one is, out of historical interest, linkage analysis is how we got to where we are today, being able to have a whole map of the human genome. It was used to localize and identify new genes for a number of important disorders. So it's got a kind of very sort of strong sort of relevance. It is sometimes used today in occasional, you do occasionally see linkage analysis applied to rare Mendelian diseases. And I guess the main thing really is it's important to have a real fundamental understanding of what's going on with linkage, because it drives into other mapping studies, association studies, and the concept of linkage disequilibrium. So it's important to have that sort of background. And another slightly different aspect of it is has a particular kind of statistics based on likelihood ratio tests, which is kind of novel and hopefully important to know. So the point of giving this lecture is really to do the kind of explaining. You can read all the stuff in a textbook and what have you, but hopefully what I want to try to do is try to take you through the things that would be a little bit tricky to pick up if you just see them written in a book. So we'll see how we all get on with that. And a few things we'll go through is we're going to talk about how you estimate something called the recombination fraction, which is kind of some sort of measure of how far apart two genetic loci are a gene from a marker or two markers or whatever it is. How you actually formally statistically test as to whether you're saying that linkage is actually happening or not, and then the sort of notion of how linkage maps were built up as a sort of skeleton framework for identifying where places were on a whole genome like the human genome. So let's begin with the historical situation and what we were faced with. We have an autosomal dominant Mendelian disorder segregating through a pedigree. The people who shaded solid have got the disease, and the other people haven't. The squares are male, the circles are female, and the idea might be that you would try to get a localization of the disease at some level of the disease gene. Whereabouts on the chromosome is it, which chromosome banded it is. You could use linkage markers to get a more refined location of some particular region of the chromosome. Then you would have to use these things called yaks and baks, artificial chromosomes, to try to get more finely spaced markers within particular regions. And eventually the ideal at the end of the day would be that you'd identify a particular change in the DNA sequence, one base change to another base in a particular gene, which disrupts that gene in a particular way, which gives people that disease. And so I've deliberately not used the sort of modern slides. I haven't updated the slides. I've kind of used the kind of slides that I've kind of inherited, and I kind of just, for example, to keep this comment in, which I nowadays find completely ironic. The guy who wrote this comment said, you'll get the complete human genome sequence and then it will be obvious. And actually, as we work more and more with whole genome sequence data, we realize that things are actually much more difficult and complicated and complex, and actually just having the whole genome sequence available is not the sort of panacea, the solution to all problems. But anyway, that's what we're going to try to do. We're going to try to go from a disease segregating in a pedigree to finding out where the disease mutation is, and then we will be able to find out what gene it's in and see what's going on and get better treatments and so on and so forth. So what do we see when we do linkage analysis? We don't see the gene, we don't see the disease mutation, we don't have no idea where it is or what it is. What we have are things that we could call genetic markers, and a genetic marker is a variation at a particular point on a chromosome that we can measure, and it could be anything. It used to be blood groups and stuff, because you know where the gene for the blood group was, and you could measure the blood group, and then you have some kind of tag as to what was going on at a particular point on a particular chromosome, as I'm going to show you. And then those so-called classical markers were replaced with DNA markers. And this was the dawn of the new genetics that occurred 1970s, 1980s, that you could take some DNA from somebody, you could do something in the lab, southern blot it or whatever, and you could tell at a particular point on a particular chromosome, does somebody have this variant or that variant? And it would be a disease, an allelic variation. Could be this bit of code, could be that bit of code, and we used RFLPs, restriction fragment length polymorphisms. But there was some kind of variation, and what the point about it was, you would know where on the chromosome it was, and you would be able to tell which allele somebody had there, and you got 2 copies of the chromosome, so they would have 2 alleles. So what you can do is you can take blood samples from these people, take out DNA, and you can have a marker at a particular point. And here we got a marker. The alleles are numbers, 5, 6, 7, whatever. So what we're going to say is that there's a particular repeat sequence at that place on that chromosome, and you can have a variable number of repeats. So it goes CA, CA, which is 2, CA, 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 3, whatever. It varies. Varies between people, varies between chromosomes. We're going to count the number of repeats, and the number of repeats is an allele, and that's variation at that point. How many repeats each chromosome would have that allele. And what we see is in the first person we got blood off, he's got 5 repeats on one chromosome, 6 on the other, the next 4 and 7, and the next 2 and 3. Those are the alleles. The 2 alleles together are called a genotype. If the alleles happen to be the same, it's called homozygous. If they happen to be different, it's heterozygous, as an aside. So that's the genotype at that marker locus. At this marker locus, halfway down the chromosome, we can see it's got 2 and 3, 1 and 5, and this person happens to have one of the chromosomes 4, and the other one is also 4, so they happen to be homozygous, or whatever. And what you notice here is that everybody's got different alleles. All these people, they're related to each other, but different alleles have come in through, and they've got different alleles. Now we get a bit closer, and watch what happens here. 2 people have now both got allele number 5. Now we don't really know if it's the same allele number 5, but they do have allele number 5. If we get here, we see that this marker, this person's got 2 copies on one chromosome, this one's got 2, and this one's got 2. So that's maybe slightly interesting. We'll come back to that. And we go here, further away from the disease gene, and here, this one's got 1, this one's got 1, this one's got something different. Okay, so that's what we've seen so far. We've just seen marker genotypes with no idea about where the disease gene is. We're trying to find it. So when we were on top of the disease gene, we did have 3 marker alleles, all the same in 3 different people. And what if we could see everybody in the pedigree and genotype everybody, look at all their DNA and see what's happening? Let's watch what happens as we trace the alleles down through the pedigree. Now, in meiosis, the chromosomes come together. We'll see in a minute, exchange genetic material, but basically each allele that we have on each chromosome, I've inherited 1 allele from my father and the other allele from my mother. Or put it the other way around, each parent has 2 alleles, and they will pass on 1 or other of those alleles to each child. So here, this ancestor, he's got a 2 and a 3, and he passes on the 2 allele to this child, passes on the 3 allele to this one, 2, 3, 2, and so on. And what we see is that each time he passes on the 2 allele, he passes on the disease, and each time he doesn't pass on the 2 allele, he doesn't pass on the disease. And that goes down through the pedigree. Every time this 2 gets passed down, it goes with the disease, 2 and disease, not 2, not disease, 2 and disease, not 2, not disease, and so on. So 2 and disease. So completely all the way through the pedigree, every time somebody inherits this ancestral 2 allele, they get the disease. Every time they don't. So this is what you see with tight linkage for an autosomal dominant disease when you have a marker that's very close to the disease gene. Now, this is one way that linkage is thought about it's a violation of Mendel's second law of independent assortment of alleles between loci. So let's imagine we have 2 heterozygote, 2 loci, 2 polymorphic loci where there are alleles. We can measure the alleles. One of the alleles we're going to call, one loci is going to be big A and little a, and another locus is going to be big B and little b. And this could be like Mendel's peas, long and tall, they're long and short, green and yellow alleles, whatever they are. We can somehow get the underlying alleles that each person has. And they're going to pass on an allele of one of the alleles that they have at the first locus to their child, one of the alleles they have at the second locus to their child. And what Mendel observed was that these 2 events would be independent of each other. Whether you got green or yellow was independent of whether you got long or short, tall or short. And that's set out in this grid here with the transmission probabilities. So if you're starting with a heterozygous parent who's got one big A, one little a, the probability of passing a big A to the child is half, the little a is a half, the probability of big B half, little b is half, and the probability of a big A and a big B together is the two, half times a half is a quarter, little a and big B quarter, and so on. So that is the law of independent assortment. Now, what you saw with that pedigree where there's linkage is that you do not get independent assortment of the disease mutation and the marker. Every time you pass on the two, you pass on the disease mutation. Every time you don't pass on the two, you don't pass on the disease mutation. So you violated that law of independent assortment. And that is, you can violate that law a lot or a little. And the idea is that deviation from complete independent assortment is what you observe when you observe linkage. And we can evaluate how often that happens. So what we can, if we think about the two alleles that we've inherited together from our father, these two different loci, and if we inherit the same combination if we pass on the same combination as we inherited, that's not recombination. If we pass on a different combination, that is recombination, as we'll see. So if you imagine that you have two variants, two variant loci, polymorphic loci which have alleles close together on the same chromosome, then, as we'll see in a moment, with meiosis, you're unlikely to have a recombination between them. So here you've got an A and a T, and we don't know from the genotype which way around they are. So we know that at one locus we've got an A and a G, and the other the C and T. So that means that they could be together. Either the A and T are together and the G and C are together, or the A is on the same chromosome as the C and the G is on the same chromosome as the T. And which way round that is called the phase of the meiosis. So here we say the A is in phase with the C and the G is in phase with the T. And we might, as we'll see later, we might or might not know what the phase is, but that is the underlying phase that the two, thinking about it in terms of chromosomes, the sort of biological underpinning of it is that the two alleles are together on the same chromosome if they're in phase, and they will tend to be inherited together as a single haplotype that will tend to go together. So a haplotype is two or more alleles which are inherited together from the same parent. Now here is the diagram of what's going on in meiosis, the physical biological phenomenon that underlies what we're kind of trying to pick up on when we do linkage analysis. We don't see this, we only see the markers, but this is the underlying biology. So let's imagine these are two chromosome 6s. I've got one chromosome 6 from my mother, one chromosome 6 from my father. I'm going to produce sperm cells, or if I'm female, I'm going to produce egg cells, and that process is called meiosis. And what happens is the first step is that each chromosome duplicates. So instead of a pair of chromosomes, I have two pairs of chromosomes, so I have 4 chromosomes. Then the chromosome exchange genetic material. So this is material that I inherited from my father and from my mother, crossing over. And now we've made new chromosomes, and the new chromosomes consist of a sort of scramble of my maternal and paternal chromosomes. But there are big chunks. There are not many crossovers, and so big chunks of the chromosome are maintained intact. And so between the B and Ds were together, and they're still together. And this diagram kind of makes it obvious that the closer together two loci are, the less likely it is that a crossover will occur between them at meiosis. And now what's going to happen is that I'm going to put one of these. Each one of these will go into a single sperm cell, so that the sperm cell will have one copy of each chromosome instead of pairs of chromosomes, and that's called haploid. And then fertilization, the haploid sperm and the haploid egg come together, the nuclei fuse, and then you have a diploid complement of chromosomes. We have a pair of each chromosome. So these are how gametes are formed. And this is the biology which underlies what we're going to try to measure when we measure linkage analysis. Now we can use, bearing in mind that quantitatively, the closer two loci are, the less likely there is to be a crossover. The further apart they are, the more likely they are to be a crossover. We can use we can count the times that we observe crossing overs as recombinations, and we can use that as an estimate of how close together or far apart two loci are. And we're going to estimate the recombination fraction. So we're going to estimate the recombination rate, and we're going to do that from the recombination fraction. We're going to observe a recombination fraction, and we're going to get an estimate of the recombination probability from that. So if you have two loci that are just on different chromosomes, then it will be 50, which is what non-syntenic means. Then it'll be 50-50 whether you get a recombination or not. So we say the recombination fraction is going 0.5. If they're together on the same chromosome, if they're syntenic, then the closer they are, the less likely it will cross over, and then we will see a recombination happening less than half the time, and the recombination fraction will be less than 0.5. And we can see how often that happens. So we don't see the chromosomes down the microscope and the genetic loci recombining. What we see is phenotypes and markers. So the phenotype is the disease. The marker will be a DNA marker that we can measure its alleles somehow or another in the lab. So here we've got a situation where we have a very unfortunate family that has two X-linked recessive diseases in it. And what that means is that males will have the disease if they inherit an abnormal gene on the X chromosome, a gene with a variant mutation in it. And females, because it's recessive, will have two X chromosomes, one carrying the disease mutation and one not. And because they've got a normal X chromosome, they will be fine and they will not have the disease. So these are two recessive X-linked diseases called Duchenne muscular dystrophy and Fragile X. And this is theoretical, but so you have these two different diseases. We're not using any markers now, we're just going to use the diseases to work out what's happening. And what we see is that some people in the family have got Duchenne's, and some people have got Fragile X. And one person, so solid, they've got both. Vertical stripes, they've got Fragile X and not Duchenne. Horizontal stripes, they've got Duchenne and not Fragile X. And no shading means they haven't got either. Now, that means we can infer what alleles they have at the disease gene. So somebody who is not shaded at all, is normal, has got a normal Duchenne gene and a normal Fragile X gene, a little d and a little f. Somebody's got both, has got a variant, a mutation in their Duchenne gene, which will put capital D, and also in their Fragile X gene, which will put capital F. And somebody who's got one, not the other, Fragile X, we put capital F, little d, and little d, the big D, little f, and so on. So just by looking to see which diseases somebody has, we can tell which copy of the gene they had. Did they get the normal one or the abnormal one? Now we know that the father was normal. He didn't have Duchenne or Fragile X, so we'll give him a little d and a little f. And we know that this mother didn't have either, so she can't have two copies of the mutation, but she's got affected sons, so she must have at least one copy. So she's got one. So we know that she's got one normal Duchenne gene and one abnormal one, and one normal Fragile X and one abnormal one. So we know everybody's alleles at these two loci. And we can look to see from this how close together is the Duchenne gene to the Fragile X gene, both on the X chromosome, how close or far apart are they? And to do that, we're going to see how many times do we get a recombination happening between the Duchenne and the Fragile X. And we're going to, so we don't know whether the two in the mother, the two are on the same chromosome as each other, the two variants, the ones causing disease, or whether on her two X chromosomes, one's on one X chromosome and one's the other. And what this is called phase unknown. So we don't know the phase of variants. We don't know if they're on the same chromosome or not, but we do know that they're there somehow. And we're going to try to make some inferences. So let's assume that we then go on and get information about the maternal grandparents. The mother's maternal grandparents means the parents of the mother. And suppose we know that they were also both phenotypically normal. So then we know that the father, the maternal grandfather didn't have anything wrong. He had normal genes. We know that the maternal grandmother must have been a carrier for both things, heterozygous at both. And we know that this mother here must have inherited the two abnormal alleles from her mother, because her father didn't have any of them. So now we know that in the mother, the two variants are in phase. They're both on the same chromosome that she's inherited, X chromosome that she's inherited from her mother. And so now we're good to make some inferences about when recombination is occurring or not, because we know the haplotypes of these two alleles in this mother. And that means that we know that this son, who has two normal alleles, there's been no recombination. He's inherited both normal alleles, so he's inherited this haplotype intact. This son has inherited this haplotype intact, both have normal alleles. He's got the other two. This son is a recombinant. He's got the Fragile X from here and the normal Duchenne from here. So there's been a crossover between the Fragile X and Duchenne in him. He's a recombinant. This one's not recombinant, not recombinant, recombinant the other way. Two normals again, not recombinant, not recombinant, and not recombinant, not recombinant, not recombinant. So we just count those up. We say there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 sons. This one and this one are recombinants. So that's 2. So 2 out of the 10 are recombinants, the rest are not recombinants. So the recombination fraction which we observe is 2 over 10, which is 0.2. So that's kind of easy, straightforward, intuitive. Recombination fraction is 0.2, and that has given us some idea about how close or far apart they were. If they were a long, long way apart, recombination fraction we'd expect to be close to 0.5. If they're very, very close together, we won't expect to see any recombination, so it should be much less than that. So this is kind of from the ground up, what we can do with a three generation, a sort of ideal 3-generation pedigree like this, is that the information from the grandparents can give us information about the phase in the parents. Then we need to have, in order to be able to see whether recombination is happening or not, both the loci, the parent has to be heterozygous at both. So what that means is that this mother has got a big D and a little d at one, big F, little f at another, and so she's heterozygous at both loci, and it's a rule of linkage that you cannot get any information about linkage from a parent unless they are heterozygous at both the loci that you're interested in. Okay, so the parent's got to be heterozygous at 2 loci, and then we can count out whether who's a recombinant and who's a non-recombinant. And I've left this formula up here. So the recombination fraction is the number of recombinants over the total number, which are the recombinants and the non-recombinants. Now, I'm putting this here because it's confusing, and you might come across this. You may see a very similar thing where instead of NR, you just have an N. So if I said there's 10 SIBs, that's N SIBs. So I might have a notation which says it's R over N, 2 over 10. Or I might have a notation which says it's R over R plus NR, 2 over 2 plus 8. And just to give you a heads up that we're going to be using that notation massively inconsistently. You'll see it in the literature inconsistently, and so you've been kind of forewarned. So very simple, recombinants over the total. And in general, if we're doing that with markers, this is a situation now where we've got, instead of a disease phenotype, we've got 2 DNA markers, and we want to know how close apart together they are or not. This is again just a very similar kind of scenario. We've got a parent here who's going to give us the linkage information. They're doubly heterozygous at 2 loci, so that's helpful. This one's homozygous at both, so she will not give any information. And we want to know the phase that these are in the parent. There's a 1, 2 and a 1, 2. We don't know which way around they are, but yes, we do, because this mother was 1, 1, 1, 1, and so both these ones must have come from the mother. That means both the 2s must have come from the grandfather, and therefore the phase in this father is the 2 1s are together and the 2 2s are together. And then when I look at the children, I can spot that this one is a recombinant, this one is a recombinant, this is not recombinant, and this is not a recombinant. Okay. cool with that. So 2 recombinants, 2 non-recombinants, recombination fraction, a half. We can also look at something about how informative individuals are, because sometimes we can make deductions and sometimes we can't. So if somebody's said, if a person is homozygous at both loci, then we know what alleles they'll pass on. So there's no information there. If somebody's heterozygous at one and homozygous at another, we know what the genotypes will be. There's or what alleles they'll get. There's a 50-50 chance they'll get a 1 and a 1, and a 50-50 chance they'll get a 1 and a 2. But we can't tell whether there's been a crossover or not, because we don't know whether this one here is the same as this one or this one. So it could have been a crossover, might not have been. We don't know if this one here is the same as this one or this one. So it could have been crossover, could have not been. So somebody who's homozygous at one locus cannot give information for linkage, forget it. On the other hand, if we have these two, then if they're in this phase, which we might know or not know, but let's assume that we know that they're in phase. This is the phase. 1, 1, 1 with a 1 and 2 with a 2. What is the probability of receiving this haplotype? Well, it's a half that you get this first one. The probability of getting that first allele is a half, because it could be this one or it could be this one. Then, if the recombination fraction is theta, we write the recombination probability as theta, probability that you get a recombination, then the probability of this one being the same as this one is 1 minus theta, and the probability of being different is theta. So the probability of getting a this one and then not a recombinant, the same one, is half for this times 1 minus theta. Likewise, for these two, it's a half times 1 minus theta. And here we've got a recombinant. What's the probability of this? It's a half that you get this times theta that you get that. Hopefully this is kind of going okay, that you have that the theta gives you the sort of conditional probability of getting one allele given that you've already got the first one. And then if the recombination fraction, if theta is a half, then the expectations for these two alleles to be inherited, these haplotypes, is a quarter, 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 which is Mendel's rule of independent assortment. So we're going to use these probabilities for observing stuff when we want to make some actual quantitative calculations of what we see. So if we, in terms of informativeness of linkage and practical issues, with a pedigree I showed you before, we could tell about the phase of people, but and we could say for sure whether somebody was a recombinant or not. Now here what we've got is we've got these grandparents telling us the phase of the alleles in the parents, and they both got a 1 in phase with a 1 and a 2 in phase with a 2. And then this person, this person, this person, and this person, we can tell how many are non-recombinant and how many recombinant haplotypes they've inherited. So this one has inherited 2 non-recombinant haplotypes, 1 from the father and 1 from the mother. This one at 2. This one has got 1 non-recombinant and 1 recombinant. And we don't know which way around it is. They've either got a non-recombinant from the father and a recombinant from the mother, or the other way around. It doesn't matter. It's still one of each. Same for this guy. This one we don't know, because it might be that they've got a 1, 1 and a 2, 2, or it might be that they've got 2 recombinants. So it could be that they've got a 1 they could have inherited a 1 and a 2 and a 2 and a 1, and we don't know that. So this person is called phase unknown, and they have either, with equal probability a priori from what we know so far, they've either got the 2 in phase or they've got the 2 recombinants. We don't know. So that's kind of ambivalence. But if you have more alleles for each marker, so here you only had 2 possible alleles, 1 or 2. That's like an RFLP or an SNP. If you have a microsatellite where you're counting repeat sequences, you might have more information, and then you can, you've got a similar situation. This guy's got a 1-1 and a 2-2. But now I've got a 3-3, and we know that the 4 and the 4 must have come from this mother, and the 3 and the 3 must have come from this grandfather, grandmother, grandfather. So we know that the 3s are in phase and the 4s are in phase. And then again, we can go through these pedigrees and we can see what's going on. But now we can follow each allele exactly. We know which has come from which parent, and we can know with greater certainty more of the time. We'll know whether somebody's a recombinant, a haplotype is a recombinant or a non-recombinant. So with people are not tending to use these highly polymorphic markers anymore, but they were useful for microsatellite linkage studies and for this kind of reason. So let's talk about a formal test as to whether linkage is present or not. And this is a kind of statistics called a likelihood ratio test, and we're going to calculate likelihoods, and we're going to formulate our hypothesis testing in terms of quantitatively by considering the likelihoods under different hypotheses. And to do that, we're going to parameterize the recombination probability. So a parameter is something that you can vary when you put into here we're going to put it into a likelihood function. We're going to have a function that defines the likelihood of our data. And we have two hypotheses. H0 is the null hypothesis, that there's no linkage. H1 is the alternative hypothesis, that there is some linkage. And that is expressed the idea that the recombination probability is something less than 0.5, though we don't know what it is. But we're going to calculate a test statistic based on the likelihoods for the likelihoods under the two hypotheses, and we're gonna it's called odds, but odds has got a different meaning in a lot of modern statistics. So it came from the term log odds. I think a modern statistician might shy away from using the term odds here, but what we're going to do is we're going to take it's a likelihood ratio, the likelihood under linkage and the likelihood under non-linkage. That's the ratio, that's this bit, which we're calling the odds of linkage. And we're going to take the logarithm base 10, as you'll see in a bit, which will just kind of make it more convenient to deal with, so we're not dealing with big numbers. And again, a modern statistician would have taken the natural logarithm of likelihood. I'll probably show you a bit later, but we're taking log base 10, and a log base 10, to remind everybody, is the power, the log of a number base 10 is the power to which you would have to raise 10 in order to get that number. So the logarithm of 100 base 10 is 2, because 100 to the power of 2, 10 to the power of 2 is 100. Log base 10 of 1,000 is 3, because 10 to the power of 3 is 1,000, and you'll see that in a second. So we're going to take the log of that, and this notation means that this is the notation for a likelihood function. So this is the likelihood for some range of values of theta between 0 and 0.5, less than 0.5. That's the likelihood under the alternative hypothesis of linkage against the likelihood under the null hypothesis of no linkage. Take those two likelihoods, calculate these likelihood functions, take their ratio, take log base 10, and that is the LOD score, which is a fantastic traditional old statistic that everybody loves to talk about, the LOD scores. Very kind of nostalgic. So that's linkage, that's likelihood and non-linkage, and we're going to take the likelihood function is what we're going to calculate now to get an actual number in there that we can plug in to this simple formula. So a likelihood is the probability of observing your data, the data that you have, if parameters have particular values, and here it's going to be if the recombination probability has a particular value or not. The probability is if something happens to be true if a certain hypothesis is true, then it's going to tell me what's the probability of observing that data. So they're kind of related. The likelihood is a probability given of a set of data that you've observed, that you've got, given a theory. You keep your data and you're going to tweak your theory, and you're going to say what would be the likelihood for me to observe this data under different theoretical conditions. And maybe I'll say at this point, this is different from a p-value. So a p-value is, I assume the null hypothesis is true. That's it. I don't know anything about any alternative hypothesis when I'm going to calculate the p-value. I'm just assuming the null hypothesis is true, and I've got a set of data, and I'll say, how unlikely would it be for me to observe this set of data or some more extreme set of data, given that the null hypothesis is true? And that probability of observing the data that you have or more extreme is called a p-value. Is not what we're doing. What we're doing here is we're going to explicitly define different hypotheses, the null hypotheses and a range of alternative hypotheses, and we're going to say, what's the exact probability for me to observe this data under each of those different hypotheses, which we will define by a parameter, and the parameter here is theta, the recombination probability, and the likelihood is the probability of the likelihood is a function of the parameter given the data that we have. And we're going to try different values for that parameter, and we're going to see the one that gives us the highest likelihood, which is intuitively the kind of best guess that we have. I'm trying to say the most likely value for that thing, but there's a whole lot of theory around likelihoods, which actually it turns out that if you want to estimate what the true value for a parameter is, the recombination probability here, then the way you do it is kind of intuitively, it's that value which means that the data that you observed is most likely. There's a particular value for the parameter that will give you the best, the most likely, the highest likelihood, and that's what we're going to find. So that's where we're going with likelihood ratio testing. So we have a likelihood ratio alternative versus null. I'm going to take the log of it as it happens, but that's basically the framework we're in. So if we had a coin, to show you how we calculate the likelihood, we're gonna say there's a coin, and that coin has a particular probability of showing up heads or not, and we don't know what that probability is. It might be loaded. It might be loaded to be usually give heads. It might be loaded to be usually give tails, and we're going to calculate the likelihood of the data that we have got, which is head, head, tail, given different probabilities for that coin to give a head. So here is the probability for the coin to give a head. If it was a normal coin, that probability is 0.5, but we think it might be a dodgy coin, so we don't know. We're going to say, what's the probability for observing the data that we have, head, head, tail, over a whole range of values for the probability to give a head. Let's see what these likelihoods we get. Now, first off, let's just note that if the probability of the likelihood under for the value that the probability of giving a head is 0, if the probability of getting a head is 0, then the likelihood is 0. We cannot, it's impossible, there's no probability at all to get head, head, tail. So that probability is 0, and the likelihood is 0 if the probability of a head is 0. And it's also 0 if the probability of a head is 1, because we have got a tail. So if the probability of getting a head is 1, we'll always get a head, then the probability of observing head, head, tail is 0, and the likelihood is 0. Okay. Now we could calculate the probability at 0.5. So what's the probability of getting head, head, tail if the probability of getting a head is 0.5? The parameter value is 0.5. So the first head is 0.5, the second head is 0.5, the third is tail is 0.5. So it's half times half times a half, which is an eighth, which is 0.125, and the likelihood of getting head, head, tail is 0.125 if the coin is fair. Now let's calculate the likelihood if, say, the probability of getting a head is, and obviously the thing it looks like is it's 2/3, doesn't it? So this is where is the maximum likelihood, and let's just take a guess that it's going to be at 2/3. So then we have 2/3 times 2/3, and then times 1/3. So the probability to get the first head is 2/3, probability to get the second head is 2/3, probability to get the third head is 1/3. And then I multiply those threes together, which is 27. 2/3 times 1/3 times 2/3 times 2/3 is 1/3. So it's going to be 4 on the top, and it's 27 on the bottom. And 4 divided by 27, well, 4 divided by 28, which is about the same thing, is 1/7. Yeah. And I happen to know that the 1/7 is 0.14 something or other. So we're in about the right place. This is 4/27 is the likelihood of observing head tail if the coin is loaded. And the coin being loaded is kind of like analogous in this context to there being linkage, that it's not 50-50, there's a recombination, that there's some value different from 50-50, and we want to estimate what the likelihood is. We can calculate what the likelihood is given different values of theta. Let's see how that works out. So what we can do is a similar graph. We can have some particular observation of how many recombinants there are, how many non-recombinants there are, and then we can graph the likelihood for that data which we observe, analogous to the head tail. We've got a certain number of recombinants, certain number of non-recombinants. We're going to specify a value for theta, which is the recombination probability going from 0 to 0.5. And by the way, every time I say recombination probability, people often just say recombination fraction. When you're talking about two, once it's established in the real world, they're kind of used interchangeably. I'm trying to use them here as the difference between what we observe and what the underlying probability is. But people tend to speak loosely about them. I'm going to try to avoid doing that. But if we can say so the recombination fraction might be taken to mean what the true recombination probability is, but either way. So what we do is we set different values for the recombination probability, going from 0 up to 0.5, and we've got 4 different data sets we've observed here from 4 different families, and each of them will have a particular curve associated with it. So let me show you what some of those curves are. Here is a curve where there are no recombinants. So if everything is not recombinant, then the maximum likelihood estimate for theta, the place where the likelihood is highest, is going to be where there are 0 recombinants, where theta is equal to 0. And that will be the place where the ratio of the alternative hypothesis against the null hypothesis of no linkage, where it has the highest value, and the logarithm of it has the highest value, the log score has the highest value, where the recombination probability is 0. And so that'll be a situation of sort of tight linkage. Here's another simple situation where everything goes negative. And this is what we observe when there are 50-50 recombinants and non-recombinants, or even more recombinants than non-recombinants. Then the maximum value for the true recombination probability, the maximum likelihood, is 0.5. We're expecting that it's random. We observe lots of recombinants. Our best fit to that data will be if the true recombination fraction is 0.5. So we'll see a curve that goes up towards 0.5, and here it will go down to infinity. If we observe some recombinants, it's impossible that the true recombination probability is 0. That can't be true, and so that likelihood will be 0, and the logarithm of 0 over the null hypothesis is the logarithm of 0 is minus infinity. So you have a curve that goes like way down and then up. This is a curve where there are some recombinants, so that goes down to minus infinity, but there are fewer recombinants than not, and in fact there's only a quarter. This is 0.25. So if we see a quarter of recombinants, then the maximum likelihood estimate for theta is a quarter. We'll have a curve that has a maximum here, and then it will go back down, and every log score is always 0 at 0.5 because that's the ratio of the null hypothesis to the null hypothesis. The ratio is 1, and then the logarithm of 1 is 0. So every log score curve like this will always go to 0 at 0.5. And the curve I haven't talked about here is this one, which is just kind of a bit weak. It doesn't go down to minus infinity. But what's happening there is there'll be some uncertainty in the data. There'll be some markers that you can't tell what the phase is. There'll be people where you don't know whether they've got the disease or not. And it's kind of like not looking good, but you've just got just not very little information at all. So while we got this up, there's a few things that are used that we talk about traditionally with log scores. One is if the log score goes over 3, then that is an indication that linkage is present. It's unlikely, we'll talk about it in a bit, but it's unlikely to get a log score of 3 by chance. And traditionally, the criterion of 3 is used to declare that linkage is present. And you might also try to say where you think the linkage is. So here, you've got a log score going up over 3, and you think the recombination fraction is probably about 0.25, but it could be somewhere, and you could take a one log score thing to make a confidence interval if you wanted to, although that's not particularly sort of formal. So log score over 3, maybe take a confidence interval. Now you got linkage, and it's probably the true recombination fraction is probably somewhere within this range. Also, you can say if the log score is negative, then there's no evidence in favor of linkage. But if you want to just say that, what range can you exclude linkage over? How close together are you pretty sure that the disease and marker are not? There, traditionally, you take a range of -2, and you say that anything within any range here that got a log score of less than -2, then the two loci are at least that far apart. So there's a log score of 3 to declare linkage is present, and a log score of -2 to declare linkage is absent. And this is a kind of traditional picture of what a 2-point log score graph looks like over a range of values of theta, the recombination probability between 0 and 0.5. Okay, so these are things that you do. Over 3, you accept linkage. -2, you say it's not in that region. If it's in the middle somewhere, then you don't really know whether linkage is there or not. And there's a notion that you can take separate families as giving independent sources of information about linkage. So you can look at one family and you might get something suggestive, but not quite enough. Now, because of the way things work, because log scores are logarithms, and they're logarithms of probabilities, and because independent probabilities multiply up together, then what you can do is you can take the log score at a particular value for theta in one family, add it to log score from another family, and you can accumulate evidence for linkage or non-linkage, and we'll see a table of that in a minute. Now, we're gonna say a few things about log scores and how you interpret them. So there's different ways of thinking about, there's different historical roots of where this log of 3 has come from. The one that I like is based on likelihood ratio statistics. So the log score of 3 is a logarithm base 10, and that is equivalent to a ratio of 1,000, 10 to the power of 3, 1,000. That the likelihood under linkage is 1,000 times higher than the likelihood under non-linkage, and that's called odds in favor of linkage of 1,000 to 1, although people don't really use that language much nowadays. In fact, nowadays we would talk about a likelihood ratio of 1,000 to 1. But it doesn't mean a p-value of 1 in 1,000, except in one very exceptional circumstance, which I might touch on later. So it's not a p is 0.001, 1 in 1,000. In fact, if you can have a p-value attached to a log score, and the way you do it is from likelihood ratio statistics, and a likelihood ratio is, if you take the typical likelihood ratio statistic is twice the natural logarithm of the likelihood ratio, and that is twice logarithm to base 2, base e. We've taken log base 10, but natural log is log base e. Twice the natural log of a likelihood ratio is a likelihood ratio statistic which is distributed as a chi-squared statistic, which means that a log of 3 is equivalent to a likelihood ratio statistic of 2 times the natural log of 10 times 3, which is about 14 or something, and that's a chi-squared statistic of about 14 with 1 degree of freedom, because you're only changing one parameter, the recombination probability, and that is so likelihood ratio statistic of 14 with 1 degree of freedom has a p-value of, it happens, 0.0001, a p-value of 301, not a p-value of 201. So people don't talk about it much, but actually a lot of 3 is equivalent to a p-value of 0.301 as it happens. So that's one way of thinking about it. Now, why have such a low p-value? Usually we use a p-value of 0.05, we use it all the time in statistics, and that's why the science is full of kind of absolutely garbage research, because we have these pathetically puny p-values, but I digress. P is 0.05. Why have P is 0.05? Why not have P is 0.05? Because you can test lots of chromosomes, you can test lots of markers, and you've got 23 chromosomes, and if you're going to do that, you're going to do tests, and it's about supposing, you had a fairly large number of tests. You want to do some sort of correction for multiple testing, and so it turns out that if you take a p-value of 301 for each of those markers, then overall, if you tested across the chromosomes, that's equivalent with multiple testing correction of about P is 0.05. So that's one way of thinking about maximum log scores. Another way is a kind of quasi-Bayesian thing where you say, what's the probability that where I look is going to be the linkage is going to be there? What's my probability of linkage? It's low. There's 20 chromosomes, 25 chromosomes, 23. It's about 1 chance in 50, probably, that if I take 2 loci at random, they're going to be linked. But then if I do a likelihood ratio test on that, then I change my prior probability of 1 in 50 to a posterior probability of 20 to 1, so it's 95 or 95% posterior probability that's probably true or not. So you can have a kind of quasi-Bayesian approach. You can have a sequential testing approach. This is the actual historical basis for this lot of 3 as set by Morton, where you would do an experiment with one set of data and see whether your log was over 3 or below -2. And if you didn't have enough data, then you do another experiment, and you do another experiment, and you do another experiment, and you keep accumulating data for or against the hypothesis until eventually your kind of overall cumulative log score either wanders over 3 or wanders under -2, at which point you have a stopping rule. And then, according to Morton, that the stopping rules that were good were a lot of 3 or a lot of -2. And then this is the one that I already mentioned, the idea of that you're going to go across the whole genome, and then a lot of about 3, or somebody said 3.3, means that once you've completed your experiment, if one of your loci, one of your markers gave a lot of over 3, then that was probably a true genome-wide significant result. Because the markers are all picking up, you can do as many markers as you like, because there's only so many different chromosomal regions, and that's very sort of equivalent to today's idea of a genome-wide significant association test when you do talk about genome-wide association studies. So anyway, it's kind of traditional, lot of 3 equals linkage. So how are we going to calculate our log scores in practice? Here we've got a pedigree, phase known, and we're going to be able to count the number of recombinants and non-recombinants. And here you see you've got N now means non-recombinant, whereas it used to be NR, I think. No, N now means total. There, I told you so. R means 2, R is 2 recombinants. P is now phase, still in phase, don't know. Anyway, they are non-recombinants, and N is the total. So the total number of recombinants is 2 out of 8. So this is how we're going to calculate the observation. So it's just standard sort of combinatorial statistics, but we're not doing any combination. We don't care about the order they're in. we're just going to say there's 2 of one and 6 of the other. And it doesn't matter what order they're in, because that's going to cancel out on the numerator and denominator for our likelihood ratio. So we're just going to calculate the likelihood as saying, what's the probability of observing R recombinants? The first one is theta, the recombination fraction, and then the second one is theta squared, third one theta cubed, and so on. We just multiply them all up, multiply the thetas together, R times. That's the probability of observing those R recombinants, and the probability of observing the non-recombinants is the n minus R, 6 in this case. The probability of observing the first one is 1 minus theta times, and then the second one, 1 minus theta. We multiply those all together, multiply them together 6 times, n minus R times. So the formula for the likelihood of a particular number of recombinants and non-recombinants is theta to the R times 1 minus theta to the n minus R. Now we're going to take the logarithm, because we're going to put this into a ratio, and we're going to take the logarithm of the ratio. So it's going to be fine if we take the logarithm now. The logarithm of this, is L theta, is the logarithm of theta times R, because you add theta up R times. When you take the logarithm of a product, then it's the sum of the logarithms of the original number. So the log of theta to the power of R is R times the logarithm of theta, and the logarithm of 1 minus theta to the power of n minus R is n minus R times the logarithm of 1 minus theta. And that is the log of the likelihood, which is what we want. If theta happens to be a half, then theta is the same as 1 minus theta, and so it's just it's n times the logarithm of a half, and that is the log likelihood at theta is a half, no linkage. So that's the log likelihood under the null hypothesis. This is the log likelihood under the alternative hypothesis. So we want to do a test, that likelihood ratio test, which I showed you before, and we got the logarithm. We're going to take the logarithm of that. It's going to give us the LOD score. This is Z is the LOD score. And when we have a ratio, the logarithm ratio is the logarithm of the numerator minus the logarithm of the denominator. So that's what we've got here. The numerator logarithm for a particular value of theta minus the log likelihood for the null hypothesis where theta is a half. And then we're going to do this over lots of different values of theta from 0 to 0.5, and we're going to see which Z gives us the highest thing. We're going to make that graph that we saw before. It would look like this, and we're going to see where it's highest. So this is the actual graph that we get when there are 2 out of 8 recombinants. So the formula is down the bottom there. It's 2 times log theta plus 6 times log 1 minus theta minus 8 times log a half. And these are the actual numbers that you get. So a few things to say about it. Let's see. Let's start with a minus infinity. Because we've got some recombinants, then the value for this at theta equals 0 is 2 times the log of 0 is 2 times the log of minus infinity, so it's minus infinity. So that goes down to minus infinity. That's fine. Here, it's if theta is a half, then this is a half, this is a half, then it's all, this is 2 times log a half plus 6 times log a half, which is 8 times log a half minus 8 times log a half, which is 0. So as I told you before, every log score at theta is 0.5 is going to be 0. And then we're going to say there's a particular point where this is highest. And the point where it's highest is, lo and behold, at 2.5. So that is the maximum likelihood, our maximum likelihood estimate for the recombination probability is the same as the recombination fraction we observed, which is 2 out of 8, 1 out of 1 quarter, 0.25 recombination probability of 0.25. And then the final thing that we'll note is that the LOD is not very high. It's only about 1. It's nowhere near, hasn't come close to this value of 3 of proving linkage, it's only slightly, mildly suggestive. We've only made 2 observations out of 6, out of 8. It's pretty, it's pretty weak. 6 out, we've made 8 observations. So what? It could easily have happened just by chance. So those are things to work out. There is one exception for the p is 0.0001 thing for a lot of 3, which is that you can get a lot of 3 from just 10 meioses. So if we imagine that we have 10 meioses and we don't see any recombinants, so recombinants are 0, then the LOD score is going to be the probability of, so we've got 0 recombinants. If the recombination probability is 0, then the probability of observing 0 recombinants is 1. We have to observe 0 recombinants. The probability of that is 1, and then the likelihood is 1, and then the log of the likelihood is 0. The log of 1 is 0. So when there are no recombinants, then at theta equals 0, the log likelihood is 0. The probability is 1 and the log likelihood is 0. And now we're going to compare that to the log likelihood under the null hypothesis. And what we've got is, it depends how we've got, supposing we've got n observations, all non-recombinant, then the log likelihood under the null hypothesis is n times log half, and it's 0, the LOD score is 0 for the alternative hypothesis minus that. So this is the formula for a LOD score when there are 0 recombinants at theta equals 0, and it turns out that it's n times 0.3. If I say n is 10, then that is a LOD of 3 from 10 non-recombinant meioses. So I see 10 in the SIP sheet, they're all non-recombinant, LOD of 3. However, the probability of observing that is not 0.00001, which is what I said asymptotically applies to a LOD of 3. In fact, you can work it out. It's 1 over 2 to the power of 10, and 2 to the power of 10 is 1,000, and 1 over 1,000 is 0.001. So in the special case that you have 10 non-recombinants, a LOD of 3 is actually only equivalent to a p-value of 0.001, but usually in a more general pedigree it's 0.001, and obviously this is all very small print. So log score analysis gets more complicated because I've been showing you perfect pedigrees and you don't necessarily know what's happening. If you don't have grandparents, then you don't know what's going on with this person. You don't know whether you've got you don't know what this phase is, so you don't know whether this is a recombinant or whether they were the other way around, and then this, so that you know whether this is recombinant or non-recombinant depending on whether it was this or this in the parent, and you don't know this one either. So you know that you're either seeing 3 non-recombinants and a recombinant, or you're seeing 3 recombinants and 1 non-recombinant, but you don't know which. So how do you calculate the likelihood of what you're observing here? Is you say it's a priori, you don't know, it could be 50-50 either way around here, and so you're going to say it's half the likelihood assuming this situation and half the likelihood assuming the other situation. Then you're going to add those together as 2 probabilities, and you get this sum. You're not labeling the meioses as the observations, haplotypes, the genotypes as recombinant or non-recombinant. You're just labeling them A and B, and you say it's half theta to the A plus times 1 minus theta to the B plus half the other way around, and that's the new likelihood. And you can put that into a sum and you can do the maths and you can get a likelihood there. You might have neither parent, and then you just see that some affected people seem to share some things, or some alleles seem to go together or not, and that's You can get that in pair analysis. And what you can do is you can set computer programs up to calculate these likelihoods with simple pedigrees, simple subsets, infinitely complicated pedigrees, bits of missing data, and so on and so forth. And you plug everything in to a program which calculates these likelihoods, and it will tell you a LOD score, and you can use that. And that is what people do in practice. Now, I think it's helpful to be aware that this was a big endeavor to set up, do likelihood these linkage calculations between pairs of markers, establish where markers were in relation to each other, how far apart they were. This was done systematically over markers. And what this was used to do was to build up a whole map of genetic markers spread out across all the human chromosomes in order that those markers could then be used for mapping studies. And this is the kind of thing that people were very proud of and would release. And you'd have markers spread across all the chromosomes, you'd say what the chromosome position was, and you would have to reconcile your recombination fraction between two markers with the actual distance that they were apart on a chromosome. And the notion was that there was a thing called physical distance, which is how many actual DNA bases they were apart, and a thing called genetic distance, which more reflected how likely it was or not that recombination would occur between them. And the measure of genetic distance, the idea is that there's some places on the chromosome where you get a lot of recombination, some places you don't get much recombination. And so the genetic distance was a way to kind of take all these estimated recombination fractions and put the markers along a genetic map. And something to say about the distance, genetic distance, is that you can't just add recombination fractions together. So if you think there's two markers and there's a recombination fraction between them of 0.2 between the first two, and then you have a third one which is also has a recombination fraction of 0.2 with one of them, it doesn't mean that the recombination fraction between the first and the third is 0.4. They don't add up that way because there's different probabilities and things will, you've got a probability of the crossover, a double crossover and so on. And so there was a way to reconcile probabilities into distances, and there were things called mapping functions. We did this called the Haldane function, the Cassambly function, and then you'd have a genetic distance which was measured, and a measure of that was a Morgan, which is a big thing. It's about almost the size of a chromosome, and then nobody used Morgans, you use centimorgans, which is a 1/100th of a Morgan, and a 1/100th of a Morgan, which was called, shown as a centimorgan, little c, big M, that's a centimorgan, is pretty equivalent to a recombination fraction of 1%. And that is a useful ballpark figure to remember, which I recommend that you remember, especially because also ballpark figure, that is about 1 megabase. So 1 million DNA bases, loosely speaking, is a centimorgan, which is loosely speaking 1% recombination fraction, recombination probability. If you have two loci which are centimorgan apart, the probability of getting crossover between them is about 1 in 100. And that's really helpful to get an idea of the resolution of linkage mapping. When we talk about linkage and we talk about association, 1 centimorgan, 1 million bases, a megabase, is a huge chunk of DNA when you're talking about genes and so on. And yet only 1% of the time will you get a crossover, and that means we'll see that means that you have to observe an awful lot of meiosis in order to get down there. And so what people did was, this is just, they built recombination maps. They had hundreds of markers spread across each chromosome, and actually the recombination distance is different in females and males because there's a higher probability of recombination males for reasons, females than males, which I don't know, and I'm sure somebody can look up. And anyway, so people built these maps, and as I say, down there, it's about a megabase, 1 million bases, 1 centimorgan. And this was how, this was how the Human Genome Mapping Project was done. This was the starting point. These lengths, these were used to map diseases, map markers, put things into the right places on the map, so that nowadays we have a human genome that we can work with. Okay, I'm going to take a break there, And then what we're going to do in the second half of the lecture is talk about how we apply all this to, especially to mapping human diseases. So hopefully catch you in part 2, and bye-bye for now.